Good morning, everyone. In the previous classes, we were discussing about eubacteria and the eubacteria's general characteristics. Then we talked about the organelles which were present inside it, such as plasma membrane. Then we talked about nucleoid. Then we talked about ribosomes. So today we are going to continue further and we are going to talk about plasmid. Plasmid is self-replicating extrachromosomal segment of double-stranded circular DNA. So what exactly we mean by this statement? When we are talking about this bacteria that we have, this is the nucleoid which is present inside and that is having the main genetic material. But apart from the main genetic material, the bacteria have extra chromosomal genetic material which is circular in shape, and that structure is called as plasmid. So plasmid has some important genes which are present inside. What exactly are genes? Genes are the segment of a particular DNA, and that segment, when it forms a protein, that segment is called as gene. So some important genes on plasmid are fertility factor, NIF genes such as nitrogen fixation genes. When we talk about the nitrogen fixing bacteria that are present out there that fix the atmospheric nitrogen, they have the NIF gene present on the plasmid of it. Then we have antibiotic resistant gene. We are going to talk about it later on. Some plasmid have the ability to join with the nucleoid temporarily and are called episomes. So in this structure, we have this arrow that is made up. So when the plasmid attaches with the nucleoid for a temporary basis, then that plasmid is called as episomes. There are plasmid that do not provide any useful trait to the cells or do not give any benefits, and such kind of plasmid are called cryptic plasmid. There are three type of useful plasmids that are there. One is F plasmid or the fertility plasmid. It is This plasmid is used for conjugation. We'll study about this later, but I'll give you a brief detail. It is primitive sexual reproduction of bacteria that we see. It is conjugation. We'll talk about it later on. So then we have R plasmid. It is called as resistance plasmid. So let's say we have this, you can say a person. This person has developed you can say staphylococcus aureus infection. So let's say we are going to give that person antibiotic penicillin to treat it. So if that person, when we know antibiotics are known to kill the bacteria, so in theoretically it should work that penicillin is going to kill that staphylococcus aureus bacteria and the infection will be gone. So but what can happen is that staphylococcus aureus bacteria, this bacteria may have this plasmid which has antibiotic resistance against this penicillin. So instead of getting killed by that penicillin, it is going to resist that damage and penicillin is not going to work, and infection will not be cleared and the person will have that infection carried on. So these kind of pathogenic bacteria that have R plasmid are very difficult to treat. So I'll give you one another example that I see that I've seen in the hospital earlier. So one of the bacteria, Mycobacterium tuberculosis, it causes the tuberculosis infection that we call as TB in a lot of places. One of the drug to treat tuberculosis is rifampicin. Nowadays what is happening is the tuberculosis bacteria, it has developed rifampicin resistance. So earlier patients were getting this treatment from rifampicin and they were getting treated, but now the resistance has been developed. So if we give rifampicin to the patients that have tuberculosis infection, rifampicin is not going to work mostly. So we have to find some another treatment, and another you can say antibiotic is provided to them instead of rifampicin. So then we have, you can say, colicin plasmid, or the col plasmid. They produce toxin colicin or bacteriocin, which are lethal to other bacteria such as enterobacterias. So when we have this bacteria, let's say we have this bacteria has this plasmid, and that plasmid has the col the colicin gene in it. When that bacteria is going to survive inside the colony that has other bacteria such as enterobacteria, this colicin gene is going to create the this col toxin, and this col toxin is going to kill that enterobacteria. So it is kind of survival of the fittest. And so that why exactly this bacteria is going to kill other bacteria is because this bacteria wants all the nutrients for itself. So it is kind of survival of the fittest kind of a theory. Then we are going to talk about the second organelle that we have here. It is inclusion bodies covered by 2 to 4 nanometer thick non-lipid proteinaceous membrane. One of the questions that I get from my students is that, sir, you have already told us that prokaryotes do not have membrane-bound organelles, but here we have this organelle inclusion bodies that does have membrane. That prokaryotes does not have membrane bound organelle. Which exact membrane are we talking about is the phospholipid bilayer membrane, but this is a very different membrane that we have. This is proteinaceous membrane, and this is the size of, this is 2 to 4 nanometer thick. So this is proteinaceous membrane. This does categorize them inside prokaryotes, and prokaryotes can have inclusion bodies. What exactly are inclusion bodies? Uh, when we say, you can say plants have vacuoles in it, and animals also have vacuoles, but they are of smaller size. So the inclusion bodies are those organelles which are a replacement of vacuole for the bacteria. So inclusion bodies are small, you can say small balloon-like structure inside the, you can say bacteria, which store different kind of components. We have gas vacuoles. Gas vacuole, as the name suggests, it stores gas inside it. It is found in cyanobacteria, purple and green sulfur bacteria. So inside, let's say we take one of the gas vacuole here, and this gas vacuole will have this hollow cylindrical vesicle which will store the gas inside it, and it will help in providing buoyancy to the bacteria. Let's say we have this surface of water here, and this bacteria is present on the surface of water, but earlier it was inside the water, but now it is daytime. So bacteria goes up with the buoyant force which is provided by these gases, which is present inside the gas vesicles, which are present further into gas vacuoles. So this is going to help the bacteria to stay on top of the water surface, and do the photosynthesis and generate its own food. Then we have inorganic inclusion, second kind of, you can say, inclusion bodies. It contains volatile granules that has inorganic phosphate inside. Iron granules is one of the type of inorganic inclusion. Then we have sulfur granules, magnetite granules. What is magnetite? It is one of the iron oxide. When magnetite granules, or you can give them another name which is called as magnetosome. It contains iron oxide inside. Why exactly the name magnetosome? Because when the bacteria which has iron oxide inside it, when those bacteria are subjected to a magnetic field, they align themselves in a particular kind of manner, or a particular, you can say, arrangement, and that arrangement is due to the magnetic field which is you can say interacting with the iron oxide present inside the magnetosome. Further, we have another point because of the ability of inorganic inclusions to pick up different colors with the basic dyes. They are also called as metachromatic granules. This inorganic inclusion and the different kind of granules, they can react with the basic dyes. There are acidic dyes, then there are basic dyes. When they this inorganic inclusion react with basic dyes, they form a color, and based upon the different kind of colors that we see, we can categorize the bacteria as well. Then we have food reserve kind of you can say inclusion body. If the food you can say is starch, Sinophysian starch, the granule is called as alpha granule. If the food is lipid based, then it is beta granule. Then if the component is protein, then we can say it is a protein granule. So this is all about inclusion bodies. Now we are going to talk about flagella. We have talked about flagellation earlier, the pattern of flagella. Now we are going to talk about the organelle flagella. So flagella has three components inside. One is basal body, one is hook, and one is filament. Basal body Basal body is few of some of the details about flagella. It is 20 nanometer in diameter and 1 to 7 micrometer in length. It is longer but a bit thicker in diameter, 20 nanometer. Bacteria flagella are made up of three parts as we have seen already. This is the picture that I took from Microbial Notes. All credits to them. So basal body is made up of rod-like structure, brings swelling in the region of plasma membrane and cell wall. There are two pair of ring in the basal body. You can say there are two pair of rings that are present inside. First, there is S and M ring. It can be this or this. I have to confirm this later on. SM ring are present inside, and the LP rings are present outside. So gram positive only have the SM ring, but gram negative have both LP and SM ring. So this is one of the detail that you have to remember. Then we have hook that we have seen. Hook is the part of the flagella that we have. It is curved tubular structure that connects filament with basal body. Basal body which was present here, this basal body is connected with the hook and the hook is connected with the filament. This curved tubular structure that connects filament with basal body, it is the thickest part of the flagellum. Then we have filament, long tubular structure that causes turbulence in liquid medium, made up of flagellin protein arranged in 3 to 8 spiral rows. So filament is made up of flagellin protein. It is present in 3 to 8 spiral rows. So you can say if we see the structure again, this filament, this is like not like this thing that you see, but it has 3 to 8 spiral rows. So this is the filament that is present there. Then we talked about flagellation earlier. We are going to just look upon that again. One type of bacteria is monotrichous. Single flagella occurs at or near the end of bacterium. Amphitrichous, a flagella at each end of the bacterium. Lophotrichous, a group or cluster of bacteria at one or both ends. Then we have peritrichous, number of flagella that are distributed all over the surface. This is the flagellation we have discussed earlier. So I just thought it would be right to just revise that once. So that is all for today. In the further classes, we are going to talk about you can say other organelles such as pili, fimbriae, and much more. Then we are going to talk about the functioning of bacteria. When we are talking about functioning, we have different type of functions such as nutrition, whether they are autotrophic, heterotrophic, saprotrophic, or you can say parasitic. We are going to talk about that. Then we'll have you can say reproduction, asexual and sexual reproduction. We'll talk more about the bacterias in the upcoming classes. So stay tuned. I hope you like the lecture, and I'll try my best to provide you more lectures in the upcoming future. Thank you.